Welcome to Azad Boy Academy. Today's lecture topic is second messenger. Today I will talk about second messenger, first messenger, and when second messenger is considered and why it is confusing with other signaling factors. Okay, so firstly, today lecture topic is second messenger. Okay, second messenger. So when I will talk about second messenger, so first messenger may come in our mind. So what is first messenger? Actually, in pharmacology or physiology, first messenger in the cellular response or cellular signaling pathway, first messenger is that which will carry the message. That means which will try to change the cellular response, will change the cellular signaling pathway. Very influential one. Okay, basically this is the extracellular one. It will produce from one cell and that will work on another cell. That is the first messenger. You can consider first messenger as a ligand. You can consider as a agonist, antagonist. There is external drug. Okay, so first messenger is, you can simply say first messenger is that which will bind the receptor. That is the first messenger. And the receptor, the first messenger has capacity to change the conformation of the receptor. So this is that will carry a message. So that will be the first messenger. And there are lots of factors which will contribute in the signaling pathway. So all of them are second messenger. Particularly, some factors can be considered as a second messenger. That is the discussion topic today. Okay, so second messenger and first messenger. So before going to the details, just I will try to give you some brief on second messenger. Second messenger, actually it has some basic criteria. That is, in the signaling pathway or cellular response pathway, some factors, some substances which will just amplify the effect, which will act very quickly, which is basically not present or present at low concentration or very basal concentration in normal situation, but upon binding the first messenger to the receptor, it will very quickly, it will produce and it will just give its action, and after completion of its activity it will destroy. Okay, so some few factors is very important. One, it must have capacity to amplify the signal, or it will just quickly influence the signaling pathway, or it will act quickly, it will diffuse from the point of synthesis to act in other place very quickly. That means it has diffusing ability and it has ability to destroy after its function. Okay, so if I just give you some example, that will be very easy for you to understand. So firstly, second messenger, I will try to classify it based on the nature. So firstly, the second messenger types, the types of second messengers. Okay, so types of second messengers. Basically, the second messengers are three types based on the nature. First one is hydro, sorry, hydrophilic. Okay, hydrophilic second messenger. You can give some example of hydrophilic second messenger, that is cyclic AMP, you can say cyclic GMP, you can say that is IP3, that is inositol triphosphate. You can think about calcium, also the second messenger, hydrophilic in nature. These are the second messenger of hydrophilic in nature. And you can say another type, that is type 2, that is hydrophobic. Okay, or you can say lipophilic. second messenger. And that the example of that will be the diacyl glycerol. You can say uh, phosphorinositidal, that means inositidal phosphate substances like PI, P2, PI, P3. You can say some arachidonic acid derivative substances, that is prostaglandin. If this is it has some controversy, these are, because it will not fulfill the whole criteria of the second messenger. Okay, and another one is sphingolipid. Sphingolipid. So these are the hydrophobic, that is lipophilic second messengers. Okay, and third one, if I just I will mention third one. You can say this is the gas in nature substance, gases. Okay, gas in nature. These are, you can say, nitric oxide, you can say carbon monoxide, you can say hydrogen sulfide. Okay, these are the second messengers. Just before going to the details, I'm just trying to introduce you to some second messengers. Okay, so these are the second messengers. So today I will try to make you clear why these are second messenger, why not others. Suppose you can think about the protein kinase A, protein kinase B, protein kinase C. You can say actin, myosin. You can say other substances which are very much important for signaling pathway, but these are not considered as a second messenger. Why? Okay, so now come to the point. Second messenger. So you have to think the 4 or 5 characteristics that will be fulfilled by the second messenger. That is, you can think in roughly, you can think uh, in generally, in your family member, anyone can carry message. Some message can carry, but when someone will just come with a important message or important person, and then a message, some quickly they will come and they will disseminate the information. Okay, so this can be like that. Just what I wanted to say, there is some second messengers are initially be present at the normal state within the cell as a basal level. That means basal level, there is low concentration normally present. Like you can think about calcium, which has normal function within your body and psychoskeletal structural matter. It has some role. You can think about diacylglycerol. It has some role to activate protein kinase C, and that will help in the cell cycle. If you think about cyclic AMP, it has some role to control some ions. So these are normally present in very low concentration, and you can say in a very basal level. Okay, so these are second messenger, but at that time it has no role as a second messenger because there is no role of first messenger. Okay, so second messenger, if you just think deeply, so first messenger will work on it and then second messenger will work. Why it is called second messenger? Just now I will make you clear. So before that, just I wanted to say first messengers may be present in the normal state within the cell in very low concentration or basal level. Okay, like that. But not all the second messengers are present in normal condition. Okay, some second messenger may be present. Okay, okay. So I will just try to make you clear. If you just think without first messenger, second messenger has no existence. So when first messenger will just bind to the receptor, the second messenger will generate. you can say just newly synthesized, or you can say newly arranged very quickly in a larger extent. So that can be considered as a second messenger. How? Come to the point. So now, second messenger, if I just give you an example, suppose if you think this is your blood vessel, so this is your endothelial layer, and this is your smooth muscle layer. Okay, so you know your in your blood vessel, if you think this is the cell membrane, and blood vessel may carry a receptor, that is M3 receptor. Okay, that is muscarinic receptor M3, and on which muscarinic receptor bind acetylcholine will bind with it. And the M3 receptor nature is like, I will not try to give you details, because another video is already present in my channel, so you can go for it, how acetylcholine causes vasodilation. Okay, so just I will try to make you understand about the second messenger. Okay, so this is the M3 receptor, and this is you can think this is your G protein. Okay, like this is your G protein, alpha, beta, gamma, and in normal condition, GDP is associated with it. GDP, like GDP. So this is inactive. After binding to the acetylcholine, this is acetylcholine, bind with the M3 receptor, then GDP will replace into the GTP. Okay, this is GTP that will be activated. So what type of G protein these are? This is G protein types is GQ. Okay, so that's activated GQ alpha part basically will activate the uh, membrane enzyme, which is called phospholipase C beta enzyme. That means activated G protein will activate the enzyme, that is PLC. Okay, that PLC will make the turn or just clip the substance which is present, like this is, you can say, PIP2, phosphorinosatidyl or 5-bisphosphate. Okay, and this PLC will just clip the PIP2 into two factor. One is DAG, another one is IP3. Okay, if you think the G protein was already present, but that was inactive before binding the acetylcholine to the receptor. PLC also present. PIP2 also present in the membrane as a component of the membrane. Okay, but DAG and IP3 is newly synthesized. Okay, so what will happen that uh, DAG will activate the PLC, sorry, protein kinase C, and that will act on gene expression like that. Okay, and what will happen? This IP3 will bind, if you think this is the IP3, and that will bind on the surface of the endoplasmic reticulum. This is the endoplasmic reticulum. You can think this is the receptor, IP3 receptor, and that will IP3 on the IP3 receptor, it will bind the IP3, and it will open the calcium channel. Okay, so I just draw here, and calcium will come out. You can think this is the calcium, because endoplasmic reticulum or sarcoplasmic reticulum are the storekeeper of calcium. Okay, then what will happen? That calcium, this is the smooth muscle, that is blood vessel. So that calcium in your blood vessel, calcium will just activate a enzyme. This is eNOS, which is present on the endoplasmic reticulum. That means over there, endoplasmic reticulum. Okay, this eNOS, that is endothelial nitric oxide synthase, not synthase, that is synthase. That will be activated by excessive calcium. Okay, so excessive calcium was not present in normal condition, which were basically present within the endoplasmic reticulum. Okay, when eNOS will be activated, then eNOS, this is the enzyme, you can think this is enzyme, and by this enzyme, eNOS will just convert the arginine into nitric oxide. Okay, this nitric oxide will produce within the endothelium of the blood vessel, and then it is the nature is gas, so it will fuse into the smooth muscle. Okay, so suppose it will go to the smooth muscle and it will just activate the membrane receptor. Here there's a membrane receptor. This is guanylyl cyclase. So this nitric oxide will just activate. It will just think this is the nitric oxide. So it will activate the guanylyl cyclase. Then guanylyl cyclase, the function of guanylyl cyclase will be GTP will converted into cyclic GMP. Okay, and that cyclic GMP will activate PKG. Okay, and that PKG will activate phosphatase enzyme. That phosphatase enzyme will dephosphorylate the myosin. That means it will just remove the phosphate from the myosin. That means it will be free of phosphate of myosin. That is free. That is inactive myosin. And that inactive myosin will unable to bind with the actin. So no actin myosin complex. No actin myosin complex. That is no contraction. That means relaxation. Okay, so if you think this signaling mechanism, then after binding acetylcholine to the M3, that is muscarinic receptor, instantly what are the substances which will produce excessive amount and that will contribute to amplify the signaling pathway? Those are the second messenger. So I will just indicate this is the second messenger, this one is second messenger, this one is second messenger, this one nitric oxide is second messenger, this one is second messenger. So these are the second messenger. Other substances like G protein already exist in active condition. PKC exists but in active condition. eNOS also was present in active condition, and that will activate quickly. So myosin, actin, all are present, but their activation is promptly changed by some particular substances which are instantly produced in a larger extent, and that will amplify quickly, and that will freeze from one part to another part, and that will work quickly in the cellular system, in the biological signal. So these are the second messengers. Okay, So this diacylglycerol is second messenger. Inositol triphosphate, excess calcium, that the calcium are present within the endoplasmic reticulum, but after the signal, then calcium excessively come out. So the calcium is second messenger, nitric oxide is second messenger, cyclic GMP is second messenger. So mostly second messengers already covered by this simple mechanism. If I just give another example by cyclic AMP, and that will cover all the second messengers. Okay, so these are the second messengers. Okay, so second messengers, the criteria already I said, it will amplify, or it may be present in lower extent in normal condition, but when first messenger will work, then second messenger will produce in a larger extent, and that will quickly work on it, and that will degrade after activation. Okay, so another example I just very quickly I will try to give you, and then I will sum up the lecture regarding second messenger. Okay, so another example, if you think this is, you can think this is your cardiac muscle. Okay, and cardiac muscle, you know, it can carry the receptor is beta 1, and you know the beta 1 is adrenergic receptor. So this is the adrenaline. Okay, beta 1, and you know the beta 1 receptor is G protein coupled receptor, and this G protein coupled receptor is GS. So GS means when G protein will be activated, the activated G protein will just activate the enzyme adenyl cyclase, and that adenyl cyclase will just convert the ATP into cyclic AMP, and that cyclic AMP will activate pKa, and that pKa will open the voltage gated calcium channel, or it can work on endoplasmic reticulum, and the calcium will enter. So excessive calcium will produce inside the cardiac cell. Then what will happen? The excess calcium, that calcium will bind. How? Suppose this is the actin, you can say this is your tropomyosin, and we can think this is troponin. Okay, calcium binding protein. So calcium will bind. If you just draw, this is the calcium. So calcium will bind with the troponin. Then what will happen? It will twist and it will make free the actin. And that is actin binding site. Actin binding site will be free. Okay, and then it will bind with the myosin. It will bind with the, when the active binding site will be free, then myosin binding site of the actin will be free. That is like that. And then myosin will bind, and then it will produce actin myosin complex, and it will causes contraction, and cardiac muscle contraction increase the heart rate, and that will increase the cardiac output, that will increase the blood pressure. Okay, so what is it? G protein previously present, but after activation of adrenaline, it will be activated, and then enzyme adenyl cyclase will be activated. So here the second messengers are, this is the second messenger. You can say this is the second messenger. So these are the second messenger. The other components are not second messenger because they are present, already exist, and after activation that will just activate the other substance, and that will assemble to each other and causes contraction. Okay, so I just give you just the basic thing of second messenger is second messenger will amplify or will just quickly increase its intensity or increase its amount to change the quick activity. Suppose if you think someone will send a message to your family, and after getting your message, then you will change the environment of your family. That means that the news can be very sad or the news can be very happy news. The environment will change. So you are the influencer, and who has sent the message? That is the first messenger. Okay, so first messenger is the drug. That is ligand, that is endogenous substance that can bind with the receptors. Okay, so mostly when second messenger you will think, so first messenger may come in your mind. Okay, some drugs, some endogenous substance has uh, uh, has the actually access to enter into the cell because they are lipophilic in nature, the molecular size is very low, so they can easily enter into the cell and even enter into the nucleus. So those are also considered as a first messenger, but second messenger is generally not connected with that. Like steroidal hormones, you can think about thyroid hormone, you can think about glucocorticosteroids. Those are also act as a fast messenger, but it has access to enter into the cell and it also enter into the nucleus. Second messenger concept is not concerned with it because first messenger enter in your house, enter in your room. So no need to arrange the second messenger. If you think in that way, that would be very easy. Okay, so first messenger bind with the extracellular receptor, that means the transmembrane receptor, also can be considered which can enter into the cell. So first messenger and second messenger. So this is all about the today's lecture. So thank you very much.